Right, this is to be going through the component 1 paper that you did as mock. Again, at your own pace and how you see as fit. If you want to pause it every now and then, you do that. If you already had an A star and you just want to look at the mark scheme for those 2-3 specific bits, then that is what you do, whatever works for you. So, no further ado, question 1. The whole question is it's kind of around the whole scenario of a practical that we did, the spinach one, and I remember a couple of groups got the different letters with the different colours in. And I know when catching up, I did it with grass with some of you as well. It's irrelevant what plant you use, you will get some photosynthetic pigments out, different compositions, and it will differ from plant to plant there. So the first question is the location, and it does say precise location. I'm actually surprised with the mark scheme there because I would not have let you get away with anything other than the thylakoid membranes. Grana, I'm finding too vague, or just thylakoid itself, I find too vague on the thylakoid membrane. So here we go. You might have been able to get away with murder a little bit there, right? That's more legible. First, the part II here. Explain the arrangement of the pigments in the photosystems. This question is asking you describe the antenna complex, so that you have got various pigments with various different wavelengths. You've got your carotenes, your xanthophylls, your chlorophyll B as the accessory pigments surrounding, bouncing the photon around until the energy is right for chlorophyll A to then absorb it, chlorophyll A being at the center of that absorption process here. Okay, so different pigments need to be mentioned. And look at this plus here. I need to see that they're absorbing different wavelengths. Okay, so different pigments by itself will not do. It needs to be a reference to the different wavelengths. You will have to have mentioned chlorophyll A at the center or the reaction center of those photosystems. And literally that the center one absorbs a slightly different wavelength. The energy needs to be right for that to be absorbed. This was a tough question to start with. So if that went a little bit wrong, do not beat yourselves up. Moving on to something that may be kind of almost a bit too simple. So it describes the whole chromatography process to you, as you did as a practical already, extracting the pigment, solvent, pencil line, blah, 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 spotting the leaf. And your actual question is why, and 2 marks for that, why is the origin line in pencil? That's your first mark. How do you get to that one? Pencil, it does not dissolve in the solvent. It will not run. It will not, like the pigments, rise. It will just stay there and do nothing. Will not dissolve in the solvent. Your second mark, why is the solvent line, literally the solvent itself, below the origin line? Think of where you are spotting. Where do you spot the leaf extract onto that origin line? So that's on the line. If the fluid level, if the solvent was actually already there, then all that spot would do, it would dissolve downwards into the fluid level. That's it. It would go from that spot downwards and just dissolve into solvent. You've got no dot or no pigments that are ready to rise. So that needs to be expressed in your answer there. And the mark scheme expresses it as pencil does not dissolve in solvent, and the pigment would dissolve and kind of run down into the solvent itself. Cool. It then moves on. The method by which the pigment is concentrated on the original line. When we did the practical, I told you that whole thing will be relatively pale unless we get another drop dried out, another drop dried out, another drop dried out to kind of just increase the local concentration in that spot. And that is what would have needed to go there. Spot it, extract it, dry it, repeat. As always, I do not look at those papers before I give them to you, so you're equally prepared for whatever's going to hit you. And the risk assessment, you just need a correct pairing here. Either the risk of the solvent to skin, and then gloves or skin protection through lab coat, or the risk to the eye as irritation, and therefore wearing goggles. So yeah, just look at that. It just needs to be the step and the precaution to go with this, the potential damage and the what do you do about it. Okay, cool. What does it say then? State one variable which you'd need to control that is used to compare the pigments in leaves of two different plants. So if you were to do the game and do spinach versus grass or spinach versus lettuce, what do you need to keep the same in order to have that same level of rising? You need to actually, the composition of the solvent itself. You remember that the hexane, propanol and acetone and alcohol kind of trying to have different combinations of solvents that work best. And I think I've described to you how long the technicians and I have tried to make this work as good as possible. So the solvent itself will need to be the same. Also the medium in which it rises in. Cheap version, that was the filter paper. Sorry, you've got the cheap version. We could do this with thin layer chromatography paper as well, which is think of it like a porous ceramic type of fluid allowed to rise in it. There are different various types of paper, different thicknesses of chromatography paper, so that would have had to stay the same. Then the little bit mathsy question, and I know I spoke to one of you who said that they actually were so anxious and nervous that they didn't see the equation right at the start. The equation is here. So I know it happens. That's what happens when you're anxious and panicking, that the most obvious things don't jump out. You've got all your numbers given for all the different pigments, and your question is here. Calculate the RF value for pigment X. Use the table, use the image, identify the pigment. So you've got pigment X, and at that point, many of you have asked for a ruler. You need to have rulers and calculators in the exam with you. Sorry, it just takes away that panic moment of, do the exam people have enough rulers and calculators spare? So ruler, calculator, remember to have that with you. Measure with a ruler from origin to your pigment front, with a ruler from your origin to your solvent front. Sometimes they're nice and they're giving you the numbers. This time they have given it to you. And then distance traveled by the pigment divided by the solvent front. That is a simple division. Give you some 0.59 something value, and then compare that to what we have got in the table here to identify, and you should have kind of gotten you to yourself to your 0.6 value for chlorophyll A. The mark scheme lets you get away with quite a range of how you're measuring it. This is in millimeters. The calculation will work out the same if you're calculating it in centimeters. And as far as I'm concerned, the mark scheme doesn't specifically say I cannot accept centimeter values. The calculation was right. You're getting to that value. That will do. Last one here. That one reminds me a little bit of a question you just did for the component 2, where you had the amino acid sequence and you have to compare an unknown amino acid sequence to a known sample. That is a standard thing we do. If you don't know something, but you know a couple of options you would have for it, compare the values for your unknown to your possible options that you've got. So if you've got an unknown, take a pure sample of the known and then compare that with the original. So I'm ignoring the question to go with this one here. The question to go with this was, you're provided with a pure solution of each pigment. Describe how you could compare, could confirm what pigment X was. So run chromatography with the pure pigments, run chromatography with pigment X, compare the RA value for X for all your known pigments. Cool, that was question 1 already.